Hey everyone, it's your instructor, Jamie Batts with A&P 2. We are continuing with our lecture on the digestive system. When we left off in the last lecture, we were talking about parts of the stomach. Now we're going to get into some of the functions of the stomach and some of the microscopic anatomy that's involved in the composition of the stomach. So, I think this is the right, there we go. Screen. This is where we left off. And so let's talk about some of the overall functions of our stomach. Obviously, this is where the chemical digestion takes place specifically through breaking down our proteins. That's the main function here of our stomach. But it's also a storage tank for ingested food. Our food can sit there for a number of hours. The food's not just chemically broken down, but it's mechanically broken down as well because the stomach is physically mixing and churning the food, which at this point, again, is called chyme. That's C-H-Y-M-E word that we mentioned last time. And specific enzymes and acids are going to work to break down the chemical structure of the chyme, of these nutrients that were in the food that we brought in. And that way, they'll be ready to be absorbed when the chyme moves into our small intestine. Intrinsic factor is also produced in the stomach, and we're going to see exactly what that is and what that does as we move through this lecture. So let's talk about how our stomach wall is put together. So first of all, we have the mucosal lining, right? All areas of our digestive tract have these major parts to them: mucosa, submucosa, muscularis, externa, serosa. So how is the composition of the stomach different from these other areas? The mucosa in the stomach has very deep folds. These deep folds form a structure called a gastric gland. It's basically like a pit in the stomach lining. There is simple columnar epithelium throughout the stomach, and this simple columnar is actually secreting a mucus to protect the epithelium itself, because there's acids and enzymes that are going to be secreted into the lumen of the stomach. So this alkaline mucus layer is there to protect the stomach lining. The gastric cells are replaced very quickly because, again, of this highly acidic, low pH environment. There's a submucosa, which has all of the blood vessels, lymphatics. The muscularis externa in the stomach has 3 muscle layers as opposed to just 2, and then the outer serosal lining. So here you can get an idea of these gastric pits, or these gastric glands. The pits are the holes themselves. The glands are inside the pits. Just a slight difference in terminology there, but you can see the highly folded ruggy of the stomach and then the deeper pits. And again, notice that they're all lined with this simple columnar. The cells differ as we go down into the pits, and we're going to take a look at how these glands are composed, the different types of cells in them. So they're going deep into the lamina propria of our stomach lining. The glands in the fundus and the body of the stomach are dominated by what we call parietal cells and chief cells. These cells are secreting acids and enzymes that are going to help move chemical digestion along. They're secreting over 1.5 liters of gastric juice every day. The glands that are within the pylorus, that lower or more distal area of our stomach, is secreting more mucus and hormones. The hormones are going to help to coordinate all of the digestive activity that goes on between the stomach and the small intestine. So, fundus and body is where parietal cells and chief cells are there. We're going to look more closely at what they do, but in general, it's acids and enzymes that they're secreting. The pylorus is secreting the mucus and hormones. So these gastric pits are the depressions that open into the interior of the stomach that form the gastric glands. There's stem cells that are within them to replace those superficial cells, right? It makes sense because not only do we have a highly acidic environment that's going to work to wear away the mucus and these cells, but you also have friction from all the food in there, right? So the pit is communicating with several gastric glands, which if you look back at this picture, you can see what we mean. So the pit is the hole on the surface, and then the gland extends deeper. So Let's talk about these cells that we mentioned. We mentioned parietal cells. Parietal cells secrete that intrinsic factor. Intrinsic factor is super important in vitamin B12 absorption. This is going to play a role later on in the digestive process, so it's kind of a preemptive secretion because B12 is not absorbed in the stomach. It's absorbed later on in the digestive process. The parietal cells also secrete HCl, hydrochloric acid. Hydrochloric acid activates pepsinogen. Pepsinogen is secreted by a different type of cell in our gastric glands, and this is to help keep the pH of our stomach very, very low. I remember this because parietal cells secrete hydrochloric acid. Hydrochloric acid begins with an H, P, H, right? Hydrochloric acid keeps the pH very, very low. So parietal cells, pH, parietal and hydrochloric acid. That's how I remember that, because there's a couple different cells that are secreting things, and you might get them mixed up. The G cells, which are in this area, these are the gastric cells. These are secreting hormones that are going to help regulate all of this and provide a feedback loop for this whole process. Chief cells are secreting pepsinogen. Pepsinogen is a chemical that's activated by the hydrochloric acid that's released from our parietal cells. So when pepsinogen becomes activated, it converts into pepsin. Pepsin is a proteolytic enzyme. What that means is it is an enzyme that's meant to break down protein. So it's going to start denaturing the protein and breaking the chemical bonds that hold the protein together in order for the amino acids to be separated out. So in infants, the chief cells are also producing renin and gastric lipase. It's not the same renin that we talked about with the kidneys. This is renin with 3 N's. I know, right? You gotta, why do they do this to us? Renin that is produced by these chief cells in infants is there to help digest milk. It's to increase the efficiency of milk digestion, as well as gastric lipase. If you recall, our salivary glands secrete an amylase, which is breaking down carbohydrates, as well as a lipase and a lingual lipase to help start the breakdown of fat. Here in our stomach, we're mostly concerned with breaking down protein with the proteolytic pepsin enzyme. Gastric lipase is also present in small quantities to continue the breakdown of fats, because fats are such a large molecule. If you remember a little bit of biochemistry, fats are so big, it takes a while to break down those hydrocarbon chains. So here you see the gastric glands and the pit. The pit itself is the opening onto the lumen. The gastric glands are the inferior part, and several glands can branch off of each pit. You have different cells, chief cells, G cells, parietal cells, all within this gland. So let's talk about how hydrochloric acid is produced. The parietal cells do not actually create hydrochloric acid in their cytoplasm, right? If the cells were actually making hydrochloric acid, then the cells themselves would be dissolving, right? It would destroy the cell. So these cells do this using the same bicarbonate chloride shift method that we learned about in our respiratory tract. So the hydrogen and the chloride are actually transported and secreted separately, and they combine in the lumen of the stomach, or in the lumen of the gastric gland, really, so that they can form hydrochloric acid. So the hydrogen ions are generated from the carbonic anhydrase, combining carbon dioxide and water together, producing bicarbonate, which dissociates into carbonic acid and a hydrogen ion. And then, just like we had in the red blood cell, that bicarbonate ion is shifted for a chloride ion, and then it's secreted together through different transport mechanisms on the cell membrane. So you have the chloride shift, and then it's pushing it right back out. So the bicarbonate is ejected into the interstitial fluid in exchange for that chloride ion, that chloride shift that we mentioned before. And then if the gastric glands are really active, the amount of bicarbonate can actually increase the pH of the blood. And that influx of bicarbonate ions is called an alkaline tide. And that could cause respiratory issues and or issues with your kidneys. Overall, the chloride ions are going to diffuse across that cell membrane, go into the lumen, and combine with the hydrogen ions, which are also actively transported into the lumen. And that's how HCl is produced. So here you see the whole process, carbon dioxide and water combining via carbonic anhydrase to form bicarbonate. The bicarbonate is dissociated into carbonic acid and a hydrogen ion, and then we have the chloride shift happening through the bloodstream. The bicarbonate goes into the blood, the chloride ion comes into the parietal cell, and then the hydrogen and chloride ion are both secreted into the lumen of the gastric gland. Step by step. Again, you can print out each of these slides individually and narrate them and or summarize them individually. Or you can print them out and see if you can put them in order and explain them to someone else. You can pause the video here, go back and review this section. We're going to keep moving on and talking about some of the specialized structures within our intestinal tract. So once we get past the stomach, the major goal now is to continue the breakdown of food and absorb the nutrients. So there's several structures that are included in our digestive tract to increase surface area. And when we increase surface area, we're increasing the ability to absorb nutrients. So there are circular folds that are basically transverse folds, kind of like imagine that the inside of your intestine is like a PVC pipe, right? So it's just this long, flat, nothing on the inside, hollow tube. Now imagine that it's a slinky, right? Not for stretching and moving purposes, but those rings of the slinky, right, have lots of folds to them. So the surface area of that compressed slinky, should have grabbed one, I got one back there. Oh well. The compressed surface area, or the surface area of that compressed slinky is much higher than the surface area of the same length PVC tube. So that's the idea behind these circular folds, just increasing the surface area. You have approximately 800 of these in the small intestine. That number is random. You don't need to know it. Most of these folds are within the jejunum section of our small intestine. So you also have intestinal villi, or villus, right? Villi and villus, so one's singular, one's plural. These are the finger-like projections on the mucosal cells themselves. They're covered by all the epithelial cells, and the epithelial cells themselves have microvilli. So the mucosa has villi, which are made of cells, and then on top of those cells are microvilli, extensions of the cytoplasm, not cilia. It's not the same thing, right? This is actual extensions of the cytoplasm and the plasma membrane into the lumen. Those are the microvilli. This is increasing the surface area hugely compared to, that flat PVC surface. We also have intestinal glands that are spread throughout. These are located typically at the base of the villi. There are stem cells that are going to divide rapidly to replace the epithelial cells that are shed from the tips of the villi, because again, anywhere where there's contact with the chyme that's passing through, there's going to be friction, so those cells are going to wear away very quickly and need to be renewed. Those cells that are shed release enzymes into the lumen, which drive these stem cells to reproduce and regenerate those new cells. So it's a feedback loop. Here you see what I mean when I talk about the folds. You have the circular folds in there, which are called the plicae circularis, right? These are those slinky rings, if you will, on the inside of our intestinal tract. Then when we look at the actual mucosa itself, you can see the projections of the mucosa, these villi going into the lumen. Then if we look at the actual, this doesn't have a microscopic picture. If we were to zoom in at each individual villi, you would see that it's made of a columnar cell that has projections on top of it as well, those microvilli. So we have huge amounts of surface area for increased potential for absorption. So the internal structure of a villus, let's kind of look back here. What we're talking about here is what is this little stack made of? What are all those pipes and tubes in there? So it's obviously an extensive capillary network, right? This is where absorption's occurring. So we have to have a highly vascularized area, lots of arteries and veins to absorb the nutrients that's coming in from our digestive lumen. So this is going to carry all of the nutrients that are absorbed through the intestinal lumen are routed through directly to the hepatic portal vein. So our superior inferior mesenteric veins are going to fuse right into the hepatic portal vein, go right into the liver. Right? We're going to get into all of the ins and outs of what happens at the liver eventually in this chapter, but that's the gist of it. All of the nutrients that are absorbed go right over to the liver for filtering, processing, kind of like data analysis. There's also a lymphatic capillary. It's a specialized lymphatic capillary called a lacteal. The lacteal, again, remember lymphatic capillaries, they're kind of these blind-ended tubes. Think of them like vacuum hoses. But in this case, it's not so much concerned with soaking up the extra interstitial fluid as it is there to soak up, I'm saying soak up, I should say absorb, to absorb the lipids that are being absorbed from your intestinal tract. So fatty acids mostly are the major component that's being absorbed through the lacteal. Why is that? Because fats don't dissolve in water, right? So if you're eating, a fatty food, and all of the glucose and amino acids, all these water-soluble compounds are being absorbed into the blood, the fatty compounds are going to be repelled, right? They don't dissolve in each other. So there's another transport mechanism there, this lacteal, that's going to allow for the transport of these fatty acids. The fatty acids are actually assembled into a kind of a protein lipid hybrid molecule, and these are going to fuse and diffuse, I should say, into the lacteal. So it's not pure fat that's going into, don't picture like big fat droplets going into your lacteals. The fatty acids are broken down and fused with a protein molecule to help make them a little bit more water soluble. Because in the lacteals, in the lymphatics themselves, it's water soluble compounds in there as well. So, and remember, all of the lymphatics ultimately drain back into the blood. So, right at the right lymphatic duct and the thoracic duct. So they're getting back into the blood no matter what. They just have to be transported differently. So again, inside the villus, we also have smooth muscle within the muscularis mucosa, right? But that's within the submucosa. This is going to help move the villi back and forth, kind of contracting it a little bit to allow for increased surface area, exposing this side and this side of the villi. So absorption can happen all the way around the villi and not just at the top. This is also going to help squeeze the lacteal, because remember, the lacteals and the lymphatics are not at the mercy of some type of pump like our cardiovascular system. So muscle contraction is the main method of movement. There's also something called a brush border on the villi. This is basically, think of it as a carpet of microvilli. Literally, picture a carpet, right? All of those fibers, the microvilli, that area, this carpet or brush border, has enzymes embedded in it, right? So this enzyme is going to help break down the rest of the substances, the nutrients that are in our chyme. So continue protein breakdown, carbohydrate breakdown, and lipid breakdown in this brush border. They're called brush border enzymes, and there's several different ones, and we'll learn about them in lab as well as here in lecture, but they contain enzymes. This brush border has enzymes for the continuation of digestion. Here you see a zoomed in view of a villi, and you can even see those simple columnar cells with the microvilli on top. You can see the capillary networks surrounding the lacteal. So again, think of it like this. The food is passing across that villi. The nutrients are being absorbed through those simple columnar cells. Then they're being transported into the interstitial fluid. Then they have to be absorbed into the bloodstream or into the lacteal. And then they're going over to the hepatic portal system to get filtered and kind of analyzed, if you will, by the liver. So I'm going to stop the video here so you have a chance to digest. I love that pun. I don't know. I'm cheesy. Digest this information, answer these 3 questions, and we'll be back in a minute, well, whenever you want, really, to go on with this next part of the lecture.